All right, so just as a reminder, this is our regular hackathon meeting. Just as a reminder, the session is also being recorded and live streamed, just like all the other sessions. So one thing that I did want to go over very quickly before we get to hackathon stuff today is tomorrow and Friday, you'll notice on the schedule that for the third session of the day after lunch, both of those days, we have breakout talks. So we have a few guest presenters coming to talk about their work that they've published using CompuCell and demonstrate their model for you. So for the Thursday sessions, we'll have two breakout sessions, one from Shiladitya Banerjee and Logan Carpenter from Carnegie Mellon University. Their topic is cell scale biophysical determinants of cell competition and epithelia. Derek Yu and Zhang Zhang will also be presenting tomorrow on the computational modeling of germinal center response and its disruption by environmental chemicals. If you would like to join either of those two talks tomorrow, the Zoom links are here. I will also send out these links in tomorrow's morning announcements email so that you can get registered for those before the talk starts. The way that that'll work is you will leave the regular session and go join those breakout rooms. And then at the end of those sessions, you'll leave those and come back to the main workshop. And I'll send out a link to the main workshop while people are in the breakout sessions, just so you have quick access to that. On Friday, we have Shane Hudson, who is from Vanderbilt, doing computational model of secondary pallet fusion and disruption. And then Mrinmoy Mukherjee, talking about cluster size distribution of cells disseminating from a primary tumor. So you have two opportunities to see two talks of published or work in progress using CompuCell, and I think that they're all going to be pretty good sessions. We may also have an additional presentation on Friday from one of our instructors, Priyam, who will be doing a discussion on cell lineage. We're still working out with the schedule if that's going to be a breakout talk on Friday or if that will be the final session for Friday. I will let you know that by tomorrow morning. And that will be also included in the announcements in the morning. And there will be an additional link for hers if there is, but I'll clarify all that in the announcements email in the morning. So make sure to keep your eyes on your inbox. So I'm not going to go through the whole spiel on the hackathon again. I think everybody's seen that enough at this point. But again, just as a reminder, here are the links. If you have not uploaded your lightning talk slides yet, please do so. And if you have not added your description to the shared Google Doc, please do so as well. We'll be taking a look at that today after the session to make suggestions for people who are looking for groups and have not found a group yet. If you have found a group through the Slack or through personal emails or anything like that, please do go into the shared Google Doc and move your name from looking for a group to which project you're in. That way we know that you've kind of decided on one already. Just to keep us from sending you a bunch of unnecessary emails about projects you might think are good if you've already decided on one. So with that, I do want to leave enough time for lightning talks. I know we have 3 people who want to present today. So we have, I think it was Norma Perez, Daniel Mari, and Liliana Houston. Daniel, did you still want to represent today?
Yeah, I can see it.
Okay, good. Does anybody else want to present today who is ready, who maybe missed the first few minutes of our intro section before? I know we have a couple of people who will be ready tomorrow. So today, I think we'll probably just stick to the 3 people doing their presentations today. And if you have questions about a model that you'd be interested in presenting tomorrow, and you have questions about the presentation you'd like to give tomorrow, we're happy to answer those during the Q&A session at the end of the day today. So I think it sounded like Norma was ready to go first there, so I'll let her take over. So again, Norma, if you would just like to go over, since we have 3 today and we have 15 minutes, I'll give you each 5 minutes since 2 of you are presenting for the first time anyway. So you can take 5 minutes and just focus mostly on the biological background, your modeling goals, and your potential teammates, and any additional details. And I'll let you know when you have about 1.5 minutes left.
Okay, thank you so much.
Yep.
May I
stop the
share?
Yes, go ahead. While she's doing that, James does say there are recorded talks for people to consider.
Yeah, can you see my screen?
Yes.
Okay, so this project is called calcium signaling at cleavage stage during zebrafish development. This is a computational approach. So the idea is to model how calcium behaves after egg fertilization. So yeah, here you are looking at an egg, and this orange color means calcium concentration. So after egg fertilization, a calcium wave is triggered that runs from the vegetal pole to the animal pole. And this activity, calcium activity, is happening together with the segregation of the ooplasm. And that produces that here starts the animal, the new animal, with one cell. And then this one cell will divide. And in this moment, the cleavage stage starts. So here, what we can see is the some intracellular propagating waves, and seems like this calcium waves comes from the reticulum. So here I have some diagram represented that part. So this is the membrane of the furrow. Here you can see the furrow, and near to the furrow, the endoplasmic reticulum is located. And seems like the calcium that is flowing or increasing here in the cytoplasm comes from these compartments, which are these compartments are the reticulum. So here I want to support that idea. So here you are looking the control case. The first cleavage has already happened here. And also then after these two cells were produced, then another division occurs, and then we finish with 4 cells. But here in this experiment, some high P3 inhibitors, the high P3 receptors are here. These receptors are these blue channels. So applying inhibitors to these IP3 receptors seems like there is a regression of this furrow. And that happened with these 2 drugs. But if some calcium channels are blocked, seems like that doesn't affect the cell division. And the other thing is that here in this experiment, already the cell division happened, but endoplasmic reticulum extracts were added to the embryo, and what happened is that a new furrow appears. So then here I want to show you how calcium behaves during that cleavage stage. So here, these are 2 cells, and you are looking in these white colors, you are looking calcium activity during this first stage of cleavage. I mean, in these first stages where cells divide. So the proposal then is to model or to couple this calcium activity to the first cell divisions. So the hypothesis is that calcium stores with IP3 receptors are the ones responsible for inducing positioning cleavage furrows via IP3 induced calcium release. And the goal is to understand how calcium is related with the cytokinesis via a computational model. And this will allow us to understand how zebrafish development takes place in the future. So
You have
yeah,
about 1 minute left, Norma.
Yeah, my modeling goals is to generate this first cleavage using CompuCell3D, coupled with a calcium signal to simulate this cleavage furrow, to test different typologies, which means to add ER in order to produce more cleavage furrow apart of the normal furrows, or to inhibit the reticulum to see what happens. So I am a postdoc at Purdue University. I have experience modeling calcium signaling in single cells. So I am looking for some team probably with experience in CompuCell3D, Python, and C, and also people from biology background regarding embryonic development. And this is my email if you are interested to join this project. Yeah, so that's it.
All right, thank you. So I think we can probably do a little bit of discussion in the chat as other people present. But let's see. So Liliana, you were also, and then Daniel will have you go after Liliana, if it's all right.
Okay, great. Let me get my slides up here. All right. How's that look?
That's good.
Great. I'm Lily, and I'm currently working on a project with two other collaborators. We met during a previous course earlier this summer, and we thought that learning CompuCell3D would be a really good contribution to our project. So we're trying to model zebrafish ray branching. So zebrafish, their tail fins regenerate when you cut them off. So as you can see, there's a cut here, and as the fin regenerates, it forms these like skeletal rays. These rays branch at a certain point. So you can see that here and here, they're branching basically like from one tube to two. And so part of the project is this whole signaling pathway that's involved in this branching, which involves sonic hedgehog. Without sonic hedgehog, we don't see any of this branching. One of my collaborators, Hannah, has been working on the biology side of this research for a couple years now. So she has really detailed schemes of those pathways, as well as the cell-cell interactions that happen because of that. So basically what's going on is there are epithelial cells moving across the osteoblasts and the pre-osteoblasts in kind of a shedding manner. And she has this, we have this hypothesis that because of upregulated the epithelial cells are adhering to the pre-osteoblasts and kind of pulling them apart into these two, into the branching ray. So there's complicated signaling pathways, but I think a good chunk to bite off for just the weekend hackathon is to just look at some of the basic cell interactions and the cell dynamics. So for example, there's this complicated scheme on the left, but on the right, we could start with just a tube of epithelial cells that over time pinch and split into two rays. And hopefully this project would continue after the hackathon, and we could hopefully build upon that and develop a more complicated model with the different cell interactions, different cell types, maybe work in signaling pathways through gradients. So I think there's a lot of potential for this project to continue and produce some interesting models that could then be compared with the experimental work that my partner Hannah is doing. So we'll skip through the wordy bits of my presentation to keep this short, but I'm currently looking for other people to work on the hackathon with me. I'm from a physics background and I'm just starting CompuCell3D this week. So someone who knows a bit more about the program would definitely be helpful, but definitely open to all backgrounds, open to someone who's interested in continuing this after as well, or not. Really, if you're interested, let me know. This is my email down here. And I did start a Slack channel for anyone who's interested. But yeah, definitely open to questions. Thanks.
Okay, great. So Ajay says, cool projects. We have maybe even one potential recruit so far, but
Awesome.
yeah, also Norma, if you want to post your email in the chat so people can get in touch with you if they're interested in your project, that would be good. And then last year we have Daniel Murray. Give me just 1 second, Daniel, to reset my timer. And whenever you're ready, you can go ahead.
Yeah, can you hear me?
Yes, you're a little bit muffled, but I can hear you.
All right. Yeah. Hi, everyone. My name is Daniel Murray. I'm a graduate student at Michigan State University. And so what we are trying to do is to model the effect of the zonal expression of TCDD activated AHR on the circadian clock genes in the liver lobe. All right. So a bit background of what we are trying to do. So the circadian clock is a molecular mechanism that synchronizes gene expression to physiology, behavior, and whatever that happens in our 24-hour cycle. And then we have the master circadian clock, that's the master pacemaker in the SCN, the suprachiasmatic nucleus, and then which synchronizes with other peripheral clocks in the kidney, liver, and others. And this circadian clock is associated with other physiological activities like temperature regulation, feeding time, rest activities. And what we want to do is to look at the toxicity of this in the liver lobe. And then looking at the liver lobe, we are looking at the spatial expression of these genes across the cells in the liver lobe. So we are looking at the expression of genes in cells that are closer to the central lobe, and then expression of genes that are in the cells that are closer to the portal lobe. Right. But there has been some studies that have shown that the expression of circadian clock genes across the liver lobe is not zonated, like their expression is equal across all the cells because of this kind of coupling that happens among these cells. But the expression of AHR, which is the ligand activator transcription factor that disrupts this circadian oscillation, is rather zonated. Its expression is zonated with higher expression in the central lobe and then lower expression in the portal lobe. And getting into a bit of the mechanism of the circadian clock. So we have the master core regulators as the clock BMAL binding to form the heterodimer, which binds to the EBOX motif to activate the circadian clock genes. And then even without AHR, we have a very nice oscillation with a period of 24 hours. And then with AHR, that is with wild type, we still have a very nice oscillation. But the moment we have TCDD, which is this environmental pollutant, which activates AHR expression, and then its activating AHR will bind to BMAL clock and then dissociate the clock from the BMAL and then disrupt the activation of the expression of the circadian clock genes. So we have an unhealthy clock here. So our goal is to theoretically model the role of this AHR in regulating the clock genes, and also to simulate the behavior and see if the zonal expression of AHR will affect the expression of the clock genes. And we have some single cell data sets that we want to fit our model to. We have a time series data set for the expression of circadian clock genes with and without TCDD. So with the model, we are building a model for circadian clock genes without TCDD, and then circadian clock genes with TCDD. And then we fit our model with this time series single cell data set to see if what they got in the experimental the experimental data is what we are getting in our model. So this project is open to anyone who is interested. So my background is in mathematics, and I started using CompuCell3D, but I'm not an expert. So anyone who is interested in the project is welcome. And then I'll post my email in the chat so that anyone who's interested can just contact me. Thank you.
Okay, great. Thank you, Daniel. All right, so like we were saying, let me just one more quick note. It's like we were saying in the chat, here are the 2 links to previous videos. If you've not had a chance to look at those yet. Daniel presented a slightly longer version of this talk in session 2, I believe it was. And those are timestamps so that you can start at the presentations if you would like to go see any of the other presentations that have been done so far. Today is probably the last time we'll keep sending you to those. I'll put them in the Slack as well, so there's a permanent link at least.